Hello, friend. Welcome to Science for Sleep. Tonight, we begin with something many people already know, even if they have never thought about it closely. Peppermint. The cool taste that lingers on the tongue. The sharp freshness in toothpaste. The quiet lift in a warm cup of tea. It is familiar, almost ordinary. And yet, beneath that simple, cool sensation, something remarkable is happening. When peppermint touches the tongue, it does not actually lower the temperature of the skin. The air does not grow colder. The body does not lose heat. Instead, peppermint contains a compound called menthol. Menthol activates tiny sensors in the nerves known as cold receptors. These receptors normally respond to real cold air or cold water, but menthol can trigger them without any drop in temperature at all. The signal travels upward, and the brain interprets it as coolness. The cooling is real in sensation, but not in physics. This small illusion is one of peppermint's defining traits. A plant capable of shifting perception without changing the environment around it. Peppermint itself is also something quietly unusual. It is not a naturally ancient species growing unchanged for thousands of years. It is a hybrid plant, formed when two other kinds of mint crossed. Spearmint and watermint met, likely in damp soil somewhere in Europe centuries ago. Their genetic material combined, and peppermint emerged as something new, stronger in aroma, higher in menthol, sharper in cooling effect. Botanists call it Mentha ex piperita. The small cross in its name marks its hybrid origin. And unlike many plants, peppermint does not reliably produce seeds of its own. It spreads underground through runners, sending out horizontal stems beneath the soil that rise again as new plants. Each one genetically identical to the one before it. Peppermint grows by extending itself quietly outward, cloning and expanding, forming wide patches of bright green leaves that release scent at the slightest brush. Cooling above the surface, spreading below it. If you enjoy these quiet journeys, you're welcome to subscribe. And if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find their way to this calm corner of the world, one sleepy traveler at a time. The quiet continues here, just as it is. Peppermint belongs to a larger family of plants known as the mint family. Many of them share similar traits. Square stems, opposite leaves, tiny oil glands on the surface of the leaf that store aromatic compounds. When those glands are crushed, the scent is released into the air almost instantly. The fragrance travels quickly because menthol and related compounds are volatile. They evaporate easily, drifting upward and outward. That is why peppermint seems to move through a room so quickly, not as a visible mist, but as invisible molecules dispersing through air. Pure peppermint oil, when cooled, can even form small menthol crystals, clear structures separating out of liquid, solid geometry emerging from scent. Even at the chemical level, peppermint expresses itself through structure and sensation. So peppermint is not simply a flavor. It is a hybrid lineage, a network of underground growth, a leaf covered in microscopic oil reservoirs, a molecule that persuades the nervous system to register cold without lowering heat, a plant that alters perception gently. And tonight, we will explore how something so small can influence breath, digestion, memory, and even the way the body interprets air moving across skin. For now, simply notice the idea of coolness, the way sensation can exist without temperature change, the way perception can shift without the world itself shifting at all. Peppermint begins there. The word mint is often used as if it means one single plant, a single flavor, a single scent. But mint is not one plant. It is a wide family of related plants, all sharing certain traits, all producing aromatic oils, all carrying some version of that cooling freshness. Peppermint is just one member of this larger group. The mint family belongs to a botanical group called mentha. Within this genus are many species: spearmint, watermint, apple mint, field mint, each one slightly different in scent and chemical balance. Some are sweet, some are sharp, some carry very little menthol at all. Peppermint stands apart because of how much menthol it contains. Spearmint, for example, has a softer aroma. Its primary compound is carvone, not menthol. Carvone produces a sweeter, gentler mint flavor. This is why spearmint tastes mild in chewing gum and cooking. It cools only lightly. It does not create that strong, icy sensation across the tongue. Watermint, another relative, grows naturally in damp soil and along streams. It contains menthol, but in lower amounts. Its scent is earthy and green, less intense. When spearmint and watermint cross naturally, likely in moist European soil, their chemical traits combined. The hybrid produced far higher levels of menthol than either parent plant alone. The result was sharper, stronger, more cooling, peppermint. In botanical terms, this kind of crossing is called hybridization. It happens when pollen from one species fertilizes another closely related species. If the genetics align well enough, a new plant form can grow. Sometimes these hybrids are weak, sometimes they are infertile, but sometimes they carry what is called hybrid vigor. Peppermint appears to be one of those cases. Its leaves grow densely, its aroma is concentrated, its oil yield is high compared to other mints. The hybrid created something stable enough to cultivate, but sterile enough that it spreads only by cloning itself. So when we say mint, we are speaking broadly. When we say peppermint, we are speaking specifically. The difference matters because chemistry matters. Even small changes in chemical composition can alter perception dramatically. A slightly higher concentration of menthol shifts the sensation from gentle coolness to sharp clarity, from soft freshness to a distinct cooling wave that seems to travel. And yet, all mints share structural similarities. Most have square stems. If you roll a mint stem gently between your fingers, you may feel its edges. Most have opposite leaves, meaning the leaves grow in pairs directly across from each other on the stem. And most contain tiny oil-producing glands that release fragrance when disturbed. These oil glands are small reservoirs, microscopic sacs storing volatile compounds. When pressure is applied, the sacs rupture. Molecules are released into the air. They travel outward quickly because they evaporate easily. That is why brushing against mint feels immediate. The scent does not linger in the leaf. It moves outward almost at once. Peppermint simply does this more intensely. Because menthol activates cold-sensitive receptors, even a small amount can shift sensation quickly. It does not need to cool the air. It only needs to reach the receptors. The larger mint family contains hundreds of varieties and cultivars developed through natural mutation and selective breeding. Some taste like chocolate, some carry hints of citrus, some are grown purely for ornamental value. But peppermint became one of the most commercially important because its chemistry aligns so well with human sensory systems. Its cooling effect is reliable, predictable, strong enough to notice but not harsh. And this distinction between mint and peppermint reminds us of something subtle. Familiar words often hide complexity. What seems like one thing is often a family of variations beneath the surface. Cooling itself exists on a spectrum. So does mint. Peppermint sits at the sharper end of that spectrum, where chemistry meets perception most clearly. From here, we move a little deeper into how this particular hybrid came to spread so widely, not by seeds carried on wind, but by quiet expansion beneath the soil, extending itself outward in ways most people never see. Peppermint did not begin as a carefully designed plant. It likely began quietly, in damp soil, where two related mints grew close enough for their pollen to mix. Spearmint and watermint flowered. Insects moved between them. Genetic material combined. And somewhere in that exchange, a new plant form appeared. That plant carried traits from both parents, but not in equal balance. Its chemistry leaned heavily toward menthol production. Its scent sharpened. Its cooling effect intensified. The hybrid had arrived. Botanists later gave it the name Mentha times piperita. The small cross in the name marks its hybrid origin. It is not a symbol of decoration. It is a marker of lineage. A reminder that peppermint is the result of two distinct species meeting. One of the most unusual things about peppermint is that it is largely sterile. Its flowers may bloom, small and pale, but they rarely produce viable seeds. The genetic combination that created peppermint also made it unable to reproduce in the traditional way. So peppermint spreads differently. Instead of relying on seeds carried by wind or animals, peppermint expands through underground stems called rhizomes. These stems grow horizontally beneath the soil. At intervals, they send up new shoots that break through the surface as fresh plants. Each one connected below. Each one genetically identical to the original. If you could see beneath the soil, you would not see separate plants. You would see a network, a web of living tissue stretching quietly outward. This is why peppermint can spread so quickly in a garden. It does not need pollination to multiply. It simply extends itself. A single plant can form a patch. A patch can become a field. Every cultivated peppermint plant today traces back to early hybrids discovered centuries ago. Because it spreads by cloning, each new plant is not a variation. It is a continuation. A botanical lineage extended forward without interruption. There is something steady about that kind of growth. No dramatic bursts, no seed pods drifting on air, just quiet horizontal expansion. Hybrid vigor may also play a role in peppermint's resilience. When two species combine, the resulting hybrid can sometimes grow more robustly than either parent. Peppermint often produces dense foliage and high oil content. It thrives in moist soil and temperate climates. Farmers harvest it just before flowering, when menthol concentration in the leaves is near its peak. Above the soil, leaves form in pairs along square stems, Tiny oil glands dot the surface. Inside those glands, menthol and related compounds accumulate as the plant grows. Sunlight drives photosynthesis. Sugars are produced. Some are converted into aromatic molecules. Cooling chemistry forms inside warm leaves. Below the soil, rhizomes continue to spread. This method of reproduction changes how humans cultivate peppermint. Because seeds are unreliable, farmers propagate peppermint by taking cuttings or root divisions. New fields are planted from pieces of existing plants. The process mirrors how peppermint grows in nature, by extension rather than by seed. There is no randomness in the new plants. They are copies, continuations of the same genetic expression. Cooling spreads in the leaf. Roots spread in the soil. Both processes are gradual. Both are persistent. When peppermint oil is distilled from harvested leaves, what is captured is the concentrated product of this quiet growth. The menthol that triggers cold receptors began as carbon fixed from sunlight. The cooling sensation begins in soil, sunlight, water, and time. Peppermint did not evolve for human breath or candy or tea. Its strong scent likely developed as a defense. Aromatic compounds can deter certain insects and grazing animals. The sharpness protects the plant. And yet, the same chemistry that discourages some creatures draws human attention. So a hybrid formed by chance became widely cultivated. A sterile plant became globally important. A network of underground growth now supports an industry that produces thousands of tons of peppermint oil each year. All of it beginning with a cross-pollination event in moist earth. A blending of two mints. A new balance of compounds. A cooling sensation that did not exist in quite the same way before. Peppermint spreads without seeds. It multiplies without visible drama. It extends quietly beneath the surface, leaf after leaf rising into air, each one holding the same genetic instructions. Cooling above, expansion below. And from that steady, unseen network, the sensation we recognize so easily begins its journey outward, from leaf to oil, from oil to receptor, from receptor to perception. The cooling from peppermint does not begin in the air. It begins in the nerves. When menthol touches the tongue or the skin, it does not lower temperature in any meaningful way. The surface does not lose heat. The molecules do not absorb warmth the way ice would. Instead, menthol interacts directly with tiny protein channels embedded in nerve endings. These channels are known as TRPM8 receptors. They are sensitive to cold. Under normal conditions, these receptors activate when the temperature of the skin drops. Cool air moving across the face. Cold water against the hands. A shift from warmth into something lower. When that drop happens, the receptor opens. Ions move across the nerve membrane. A signal begins. The brain receives that signal and interprets it as cold. Menthol can open those same receptors. It binds to the channel and causes it to activate even when the surrounding temperature remains unchanged. The receptor responds as if the environment has cooled. The signal travels along the nerve. The brain interprets it the same way it would interpret actual cold air. The cooling is perceptual. This is why peppermint can feel cold even in a warm room, even in warm water, even when placed on skin that remains physically unchanged. The sensation can spread gradually, a light touch at first, then a widening awareness. Receptors activating across the tongue or lips. The signal does not remain localized. The nervous system integrates it into a broader sensory map. Coolness seems to move, even though the molecule itself does not travel very far. It is not the movement of menthol that spreads. It is the activation of receptors in sequence. Menthol does more than trigger cold sensation. At slightly higher concentrations, it can also reduce the perception of discomfort. TRPM8. Activation can influence pain pathways by modulating how certain nerve signals are transmitted. The cooling sensation competes with other sensory inputs. In simple terms, when the brain receives a strong cold signal, it may pay less attention to minor irritation or soreness in the same area. This is part of why menthol appears in muscle balms and topical creams. The effect is not numbing in the way anesthetics work. It does not shut down sensation completely. Instead, it shifts attention within the sensory system. Cooling moves to the foreground. Other signals soften. This same receptor system exists in the nasal passages. When menthol vapor is inhaled, it stimulates cold-sensitive receptors inside the nose. The brain interprets this as increased airflow. Yet menthol does not physically widen the airways. It does not reduce swelling directly in the way a decongestant might. It changes perception. The air feels clearer because the receptors signal coolness, and coolness is often associated with open air moving freely. The body reads the sensation and translates it into a feeling of easier breathing. This interplay between molecule and receptor is precise. TRPM8 channels are part of a larger family of temperature-sensitive proteins that help the body maintain awareness of its environment. Some respond to heat, others to mechanical pressure, others to chemical stimuli. Menthol happens to align perfectly with one of them. A plant compound fits into a human receptor, and from that alignment, sensation changes. Cooling from peppermint does not require wind. It does not require winter. It does not require shade. It requires only a small concentration of menthol interacting with the right molecular structure. As receptors activate, signals travel through peripheral nerves into the spinal cord and upward toward the brain. Interpretation happens quickly. The brain does not distinguish between cold caused by temperature and cold caused by menthol binding. The electrical signal is similar enough that perception feels real. In this way, peppermint demonstrates something subtle about the sensory system. Experience is constructed from signals. Signals arise from receptors. Receptors can be activated by more than one kind of input. A drop in temperature, or a molecule shaped just right. The cooling sensation lingers briefly, then fades as receptors adapt. TRPM8 channels reduce their activity after sustained stimulation. The brain recalibrates. What felt sharply cool becomes softer, less distinct. Perception adjusts. Peppermint does not change the room. It changes signaling. It does not cool the air. It cools awareness. And from that simple molecular interaction, a familiar sensation emerges again and again, on the tongue, in the breath, across the skin, not as a drop in temperature, but as a quiet illusion shaped by chemistry and nerves. The cooling sensation from peppermint begins at the surface, but its effects do not end there. Once menthol activates cold-sensitive receptors, the signal does not simply stop at the skin or tongue. It travels inward along nerve pathways, moving toward the central nervous system, where it is interpreted, integrated, and sometimes softened. Menthol interacts not only with TRPM8 cold receptors, but also with other channels involved in sensory signaling. Some of these channels help regulate how strongly nerve cells fire. When menthol binds to certain receptors, it can reduce the intensity of electrical signals traveling through peripheral nerves. The result is subtle: a slight dampening of sharpness, a mild decrease in perceived irritation. This is one reason menthol is described as having analgesic or pain-relieving properties. It does not block pain completely. It does not silence nerve endings in the way stronger medications do. Instead, it shifts the balance of signals. Cooling input rises to the foreground. Minor discomfort recedes slightly into the background. The nervous system prioritizes one sensation over another. The body is constantly sorting sensory information: temperature, pressure, movement, irritation. Each signal competes for attention. When menthol activates cold receptors, it adds a strong but non-threatening stimulus into the system. The brain interprets it as cooling, and that interpretation can gently override weaker signals nearby. This process is sometimes called counter-irritation. A mild, non-harmful sensation can reduce awareness of a deeper or dull ache beneath it. When menthol is applied to skin in muscle balms, the cooling sensation draws attention outward. Blood vessels near the surface may dilate slightly in response to the stimulus, creating a feeling of warmth after the initial coolness fades. Cooling, then gentle warmth. Alternating perception. The nervous system responds to contrast. Menthol also influences calcium channels within certain nerve cells. Calcium ions play a role in how signals are transmitted between neurons. By interacting with these channels, menthol can subtly modulate the flow of information along nerve pathways. The effect is mild but measurable. Sensory transmission becomes slightly altered. Because of these properties, menthol appears in products designed to ease tension headaches. Applied to the temples or forehead, it produces cooling across the skin. Studies have shown that topical menthol can reduce headache intensity in some individuals, particularly when used early. The mechanism is not fully understood, but the sensory shift appears to interrupt the escalation of discomfort signals. Peppermint's influence on the nervous system extends beyond the skin. In the digestive tract, menthol interacts with smooth muscle and the nerves that regulate contraction. The intestines are lined with muscle fibers that contract rhythmically to move food along. These contractions are controlled by both local nerve networks and the autonomic nervous system. Menthol can help relax these smooth muscles, reducing spasms. The effect is gentle. In clinical studies, peppermint oil has been shown to reduce symptoms in individuals with irritable bowel syndrome. The relaxation of intestinal muscle may ease cramping and discomfort. Enteric-coated capsules are often used in research to ensure that peppermint oil passes through the stomach and dissolves in the intestines, where it can act directly on smooth muscle tissue. This interaction again reflects a pattern. Peppermint does not overpower the system. It nudges signaling. It adjusts tone. The nervous system is built to respond to temperature changes, chemical signals, and mechanical pressure. Menthol fits into that system in a precise way. It does not introduce something foreign. It activates pathways that already exist. It engages receptors designed to interpret environmental change. Cooling becomes a message, and that message travels through sensory neurons, up toward the spinal cord, and onward into the brain, where perception forms. The body remains at the same temperature. The muscles remain intact. The environment remains unchanged. Yet signaling shifts. That shift is enough to alter experience. Peppermint interacts with nerve endings, with ion channels, with smooth muscle tissue. It influences how signals are sent, how they are prioritized, and how they are interpreted. Forcefully? No. Dramatically? No, but consistently. Cooling spreads across receptors. Signals move inward. Perception adjusts. The nervous system responds to what it senses. And peppermint offers it a sensation that feels clean, clear, and slightly softer than what came before. Cooling begins at the surface, but peppermint's influence often travels inward, toward one of the body's most steady and continuous systems. The digestive tract moves quietly throughout the day, contracting and relaxing in waves that push food forward. These movements are controlled by smooth muscle, a type of muscle that works automatically without conscious effort. Smooth muscle is different from the muscles in your arms or legs. It does not contract sharply on command. It contracts slowly, rhythmically, guided by nerve signals and chemical messengers. In the intestines, these contractions form a pattern called peristalsis, a gentle squeezing motion that moves contents forward. Peppermint interacts with this system. Menthol and related compounds in peppermint oil can relax smooth muscle tissue. They do this by influencing calcium channels within the muscle cells. Calcium ions play a central role in muscle contraction. When calcium enters the cell, contraction strengthens. When calcium flow is reduced, the muscle relaxes. Menthol appears to help reduce the intensity of these contractions in certain contexts. This relaxation can be especially helpful when the muscles contract too strongly or irregularly. In conditions such as irritable bowel syndrome or IBS, intestinal muscles may spasm or tighten unpredictably. These spasms can create cramping, bloating, and discomfort. Clinical studies have shown that peppermint oil can reduce symptoms of IBS in some individuals. In several controlled trials, patients who took enteric-coated peppermint oil capsules reported less abdominal pain and fewer episodes of cramping compared to those taking a placebo. The enteric coating is important. It prevents the capsule from dissolving in the stomach, where concentrated oil might cause irritation. Instead, the capsule passes intact into the intestines before releasing its contents. There, the peppermint oil comes into direct contact with the smooth muscle lining the digestive tract. The effect is not dramatic. It is not a complete reset, but it can soften the intensity of contractions. It can reduce the sensation of tightness. Gas-related discomfort may also lessen when muscle spasms ease. When the intestines contract too forcefully, gas can become trapped, stretching the walls and creating pressure. Relaxation allows movement to resume more smoothly. Again, peppermint does not remove gas chemically, It does not alter digestion at the level of nutrients. It alters signaling. It influences how muscle tissue responds to those signals. The digestive system contains its own network of nerves, sometimes called the enteric nervous system. This system operates semi-independently, regulating movement and secretion within the gut. Peppermint's compounds can interact locally within this network, adjusting tone and rhythm. Cooling receptors are present here as well. TRPM8 channels exist in parts of the digestive tract. When activated, they may contribute to the perception of soothing coolness internally, even though temperature does not change in any meaningful way. So the sensation of peppermint tea settling the stomach may arise from both mechanical relaxation and altered perception. Warm liquid enters the stomach. Volatile compounds disperse. Smooth muscle tone shifts slightly. Signals adjust. The experience becomes softer. Peppermint tea has been used for centuries after meals. The ritual itself is gentle. Steam rising from the cup carries aroma upward before the liquid even touches the lips. The scent prepares the sensory system. The warmth supports circulation. The compounds dissolve gradually into the digestive tract. Relaxation unfolds not as a dramatic event, but as a series of small shifts. Muscle fibers loosen. Signals quiet slightly. Pressure eases. Peppermint does not stop digestion. It does not silence the system. It helps recalibrate tension when it becomes excessive. Inside the body, contraction and release. Continuing quiet cycles. Peppermint participates in those cycles through chemistry and receptor interaction. Cooling at the surface, softening beneath it. And from this internal settling, attention can drift upward again toward the breath, where menthol once more meets receptors designed to interpret air moving across tissue. Air moves constantly through the body, in and out, in and out. The lungs expand, the chest rises, the diaphragm lowers and lifts again. Most of the time, this happens without awareness. Breath continues quietly in the background. When peppermint enters that process, something shifts. Menthol does not increase the amount of oxygen entering the lungs. It does not physically widen the airways in a lasting way. Yet many people describe a sensation of clearer breathing after inhaling peppermint. That sensation begins in the lining of the nasal passages. Inside the nose are cold sensitive receptors, the same TRPM8 channels that respond on the tongue and skin. When menthol vapor reaches them, they activate. The brain receives the signal and interprets it as cool air, moving through the nasal cavity. Cool air is often associated with open airflow. The body reads the signal and constructs a perception of ease, even though the diameter of the airway remains unchanged. This is why menthol appears in cough drops, inhalers, and vapor rubs. The compound does not remove mucus directly. It does not eliminate inflammation, but it alters perception in a way that feels relieving. Air seems to pass more smoothly. Peppermint oil contains volatile molecules, meaning they evaporate easily at room temperature. When a peppermint leaf is crushed or a drop of oil is exposed to air, these molecules lift and disperse. They travel quickly because they are light and highly aromatic. As they enter the nose, they bind to olfactory receptors located high in the nasal cavity. These receptors send signals directly to the olfactory bulb, a structure at the front of the brain. The olfactory bulb has a direct connection to the limbic system, which is involved in memory and emotion. Unlike other senses, smell does not pass first through the thalamus for processing. It has a more direct route inward. This may be one reason scent can feel immediate and vivid. Peppermint's aroma is sharp and distinct. It cuts through background smells. It resets attention briefly, not because it cleans the air chemically, but because it stimulates receptors strongly enough to dominate the sensory field. Mint does not neutralize odors at a molecular level in most contexts. It masks them. It overlays its own signal on top of other signals. The brain prioritizes the stronger input. Freshness, then, is not the absence of scent. It is the presence of a different scent. Over time, the receptors adapt. When exposed continuously to menthol, TRPM8 channels reduce their response. The intensity fades. What felt sharply cool softens into neutrality. This adaptation is a built-in feature of the nervous system. It prevents overstimulation. It allows new signals to stand out. Peppermint feels most intense at first contact, a cool rush, a clear line of sensation moving inward through the nose and throat. Then it settles. Breathing becomes steady again. The contrast between before and after is what makes the effect noticeable. When peppermint tea is lifted toward the face, vapor rises before the liquid touches the lips. The scent reaches receptors first. Breath carries volatile compounds inward. The lungs continue their rhythm. Menthol does not change the composition of the air significantly. Oxygen and carbon dioxide exchange proceed as usual in the alveoli, the tiny air sacs deep in the lungs. Gas exchange remains governed by diffusion and concentration gradients. But perception changes. Air feels lighter, cooler. The body reads that as openness. Peppermint's interaction with breath demonstrates once again that sensation is constructed. The nervous system interprets signals based on receptor activation, not on direct measurement of airflow volume. A small molecule fits into a channel designed for cold detection. The signal moves upward. The brain assembles meaning. Clear, open, fresh. The air has not transformed. The receptors have responded. And from that response, breath feels slightly different. Cooling does not need to alter the atmosphere to alter experience. It only needs to reach the right receptors at the right moment, carried gently on moving air. Long before peppermint reaches a cup of tea or a small glass bottle, it exists as something dispersed inside a leaf. Tiny oil glands sit along the surface, especially on the underside, each one holding a concentrated mixture of volatile compounds. These glands are called trichomes. Under magnification, they appear like small translucent domes. Inside them, menthol and related molecules accumulate as the plant grows. When the leaves are harvested for oil, the goal is to gather those compounds without burning or degrading them. Peppermint oil is typically extracted through steam distillation, a process that uses heat and vapor to separate volatile molecules from plant tissue. The leaves are placed into a distillation chamber. Steam passes upward through the plant material. Heat causes the oil glands to rupture. The volatile compounds evaporate along with the steam. This mixture of vapor and oil rises and then moves into a cooling chamber, where it condenses back into liquid form. Because oil and water do not mix, they separate naturally. The peppermint oil floats above the condensed water, which is sometimes called hydrosol. The oil layer can then be collected. The entire transformation depends on volatility. Peppermint's compounds evaporate at relatively low temperatures compared to many other plant molecules. That is why steam, rather than direct boiling, is used. The process protects the structure of the oil. The major compounds in peppermint oil include menthol, menthone, and menthol acetate. Menthol is responsible for the cooling sensation. Menthone contributes a sharp, slightly herbal scent. Menthol acetate adds a softer, slightly sweet note. Together, they create the balanced aroma that people recognize instantly. The concentration of menthol in peppermint oil is much higher than in the fresh leaf. In raw leaf form, menthol is diluted within plant tissue and water content. In oil form, it becomes concentrated. A few drops contain the aromatic power of many leaves. Temperature affects this process at multiple stages. The growing climate influences how much menthol the plant produces. Cooler growing seasons often increase menthol concentration. Harvest timing also matters. Farmers typically cut peppermint just before flowering, when oil content in the leaves reaches its peak. Even after extraction, temperature continues to shape peppermint oil's behavior. Menthol has a relatively high melting point compared to many other volatile plant compounds. When peppermint oil is cooled, menthol can crystallize. Tiny clear crystals form within the liquid, especially if the oil is pure and highly concentrated. This crystallization does not mean the oil is damaged. It reflects menthol's physical properties. If warmed gently, the crystals dissolve again. In that small shift from liquid to solid and back again, peppermint reveals another aspect of its structure. The cooling compound that activates receptors on the tongue also has its own thermal behavior. Oil yield varies depending on cultivation methods and environmental conditions. Peppermint grown in the Pacific Northwest of the United States is known for high menthol content. India also produces large quantities of menthol-rich oil. Soil moisture, sunlight exposure, and harvest timing all influence the final chemical profile. The chemistry remains precise. Even small variations in menthol percentage can alter how strongly the oil feels when applied or inhaled. Once bottled, peppermint oil continues to release vapor slowly when exposed to air. The volatile molecules rise. They move outward invisibly. They reach receptors and activate the same cold-sensitive channels described earlier. Inside the leaf, the compounds are stored quietly. Through distillation, they are gathered. In oil form, they become concentrated. Steam rises, vapor condenses, oil separates. The transformation is simple in principle, but careful in execution. What began as sunlight captured by chlorophyll becomes a clear liquid, capable of shifting perception through chemistry alone. Menthol dissolves in oil. Oil disperses in air. Air carries molecules toward receptors. And from that distillation process, the familiar sensation of peppermint emerges again, now intensified and ready to travel beyond the leaf that first formed it. From chemistry, we begin to move outward into time toward the ways humans first encountered this plant and folded it into daily life. Before peppermint oil was measured in glass bottles, Before steam rose through metal distillation chambers, mint leaves were gathered by hand. Their scent was noticed long before their chemistry was understood. People brushed against the plant and felt its coolness. They crushed the leaves and released fragrance into the air. The experience came first. Explanation came much later. Mint appears in ancient records across several early civilizations, while peppermint itself as a named hybrid emerged later. Wild mints were known and valued thousands of years ago. In ancient Greece, mint was associated with hospitality. Leaves were sometimes rubbed across wooden tables before meals. The scent spread lightly through the space, not to disinfect in a modern chemical sense, but to refresh, to prepare the senses for food and company. The Greeks also told a story about a nymph named Mintha. According to myth, Mintha caught the attention of Hades. Persephone, jealous, transformed her into a low-growing plant that would be stepped on. But Hades softened the punishment by giving the plant a sweet and strong fragrance. Even when crushed underfoot, mint would release scent into the air. In that story, scent becomes resilience. Even pressure releases fragrance. Even transformation leaves something lasting. Mint was not only symbolic, it was practical. The Romans used mint in sauces and wine. They mixed it with honey and vinegar. Roman naturalist Pliny the Elder wrote about mint's ability to stimulate the appetite. Mint wreaths were sometimes worn at feasts, the scent rising as people moved. In these uses, mint became associated with alertness and clarity, a fresh sensation before and during eating. Archaeological evidence suggests mint may also have been present in ancient Egypt. Dried mint leaves have been found in tomb sites. Herbs were often included in burial practices, sometimes for symbolic meaning, sometimes for preservation, sometimes for fragrance. The air in enclosed spaces can grow stale. Aromatic plants offered a way to soften that effect. The scent of mint would have been noticeable in close quarters. Across early cultures, mint became linked with renewal. The scent cuts through heavier smells. It rises quickly. It feels clean, even if cleanliness was not chemically verified in the way modern science would define it. The cooling sensation may also have contributed to its use in warm climates. In hot regions, a plant that triggers cold receptors offers a form of perceptual relief. Even if the air remains warm, the signal suggests something cooler. Mint does not lower the temperature of a room, but it can lower the perceived heat of experience. In ancient medicine, mint was used to ease stomach discomfort. Herbal texts from Greece and Rome describe mint infusions for digestive complaints. The language was different then. Smooth muscle and calcium channels were not known, but the pattern of observation was similar. Mint seemed to calm certain internal tensions. Over time, different mint species traveled along trade routes. Seeds and cuttings moved between regions. What began as local plants became shared resources. The scent of mint began appearing in more places. In each culture, mint was interpreted slightly differently, as hospitality, as healing, as appetite stimulation, as fragrance for the dead, as a symbol in myth. The plant itself remained the same in structure. Square stems, opposite leaves, oil glands holding volatile compounds. But the meaning layered onto it shifted. Cooling sensation met cultural story. What connects these early uses is sensory experience. People responded to what they could feel and smell. They did not measure menthol percentage. They did not name receptors. They noticed effects. The scent traveled through air. The cooling touched the tongue. The leaves were gathered and dried. Peppermint, as a specific hybrid, would later become more defined and widely cultivated. But mint in general had already secured its place in daily life long before botanical classification. From tables scented before meals to myths explaining crushed leaves, mint became part of human ritual. The fragrance rose in warm rooms. The leaves were steeped in water. The cooling sensation repeated across generations. From these early practices, mint would move forward into monastery gardens and medieval apothecaries, where cultivation became more deliberate and written records began to shape how the plant was understood and preserved. As mint moved through ancient cultures and into later centuries, it found a steady home in monastery gardens. During the medieval period, monasteries were centers of learning, medicine, and agriculture. Monks cultivated herbs not only for food, but for healing. Garden plots were carefully organized, each plant grown for a specific purpose. Mint, including early forms of peppermint, was among them. These gardens were practical spaces, raised beds bordered by stone, soil turned by hand, plants labeled in Latin. The air would have carried the scent of crushed leaves as herbs were gathered for use. Peppermint, once recognized as a distinct hybrid in Europe, became especially valued for its strong aroma and digestive properties. Medieval herbal texts recorded observations passed down through generations. Mint was recommended for stomach discomfort, for easing nausea, and for clearing the head. The language of the time described humors and balances rather than receptors and muscle relaxation, but the experiential patterns were similar. Peppermint tea, or mint infusion, was prepared by steeping leaves in hot water. The warmth released volatile oils. Steam carried the scent upward before the liquid was even tasted. The act of drinking became both medicinal and ritual. Respiratory uses were also common. Mint leaves were crushed and inhaled. The cooling sensation in the nose may have created the impression of clearer breathing, just as it does today. Even without modern knowledge of TRPM8 receptors, the sensory experience guided usage. Apothecaries began to distill plant oils during this time. Early distillation equipment was simple. Glass or ceramic vessels heated gently over flame. Vapors collected and condensed. Essential oils were more concentrated than leaf infusions and therefore used carefully. Peppermint oil, when available, would have been considered potent. The development of distillation techniques marked an important shift. Instead of using the whole leaf, practitioners could isolate the aromatic fraction. Menthol-rich oil could be applied directly in small amounts. Trade routes across Europe and into Asia expanded the availability of herbs. Peppermint and other mints traveled in dried bundles. Seeds and cuttings were exchanged. Botanical gardens began cataloging plant varieties more systematically. Peppermint's hybrid nature may not have been fully understood at first, but its distinctive cooling quality set it apart. In England, peppermint cultivation became more organized by the late 17th century. It was formally described in botanical literature during that period. Fields of peppermint began appearing in damp regions suitable for its growth. The plant's preference for moist soil and moderate temperatures made parts of Europe ideal for cultivation. Medicinal manuscripts from the medieval and early modern periods often included mint among recommended herbs for common ailments. Headaches, digestive discomfort, bad breath. The uses were consistent across regions. Peppermint became woven into daily routines. A sprig might be chewed after meals. Leaves might be placed in water basins to freshen hands. Dried mint might be stored in cloth bags to scent rooms. Because peppermint spreads through rhizomes rather than seeds, farmers learned to divide roots and replant sections to expand fields. This method mirrored the plant's natural growth pattern. Cultivation followed biology. The cooling effect remained central to its identity. In an era without refrigeration, the perception of coolness carried psychological weight. A cooling sensation could feel refreshing in warm weather. It could feel clarifying during fatigue. Peppermint's strong aroma also made it useful in environments where sanitation was limited. It masked unpleasant odors. The volatile oils dispersed quickly in air. The scent dominated perception. Monastery records and herbal books preserved knowledge about peppermint's uses. These texts served as early medical references, guiding treatment in communities that had limited access to physicians. From garden bed to manuscript page, peppermint transitioned from wild plant to cultivated resource. Its identity became more defined, its properties more deliberately applied. As trade expanded and knowledge accumulated, peppermint moved beyond monastery walls and into broader society. Its leaves continued to meet hot water. Its scent continued to rise into air. And from those quiet medieval gardens, peppermint would eventually enter commercial production, refined extraction methods, and the growing scientific frameworks that would later explain its effects in molecular detail. Leaves meet water, and something begins to move. When dried peppermint leaves are placed into hot water, the heat does not simply warm them. It loosens what has been stored inside. The tiny oil glands along the surface of the leaf soften. Volatile compounds begin to release. Steam carries fragrance upward before the liquid itself is tasted. The transformation is gradual. Infusion is different from distillation. No oil is separated out. No steam is captured and condensed. Instead, water acts as a solvent, drawing certain compounds from the leaf into the surrounding liquid. Some of peppermint's components dissolve in water easily. Others are less water-soluble, but still disperse enough to flavor the infusion. Temperature matters. If the water is too cool, fewer aromatic compounds are released. If the water is boiling aggressively, some volatile molecules may dissipate into the air before they dissolve. Most recommendations suggest steeping peppermint leaves in water that has just reached a boil, then allowing them to sit for 5 to 10 minutes. During those minutes, diffusion takes place. Molecules move from areas of higher concentration inside the leaf to lower concentration in the surrounding water. The leaf tissue softens. The liquid deepens slightly in color, often turning pale green or light amber. The scent rises first. Olfactory receptors activate before taste receptors do. This is part of why peppermint tea can feel calming even before the first sip. Breath carries the vapor inward. Menthol stimulates cold-sensitive receptors inside the nose. The brain registers coolness layered over warmth. Then the liquid touches the tongue. Menthol dissolves into saliva. TRPM8 receptors activate along the surface of the mouth. Cooling spreads across taste buds. Yet the tea itself is warm. Heat and cool sensation coexist. The nervous system interprets both signals simultaneously. Warmth from temperature, coolness from chemistry. The digestive tract receives the liquid next. Warm fluids can help relax the stomach slightly by increasing circulation. Peppermint compounds move with the liquid downward. Smooth muscle may respond gently to menthol's presence, easing minor tension. Clinical research has examined peppermint tea and peppermint oil for digestive symptoms. While oil capsules are often used in formal trials because of dosage precision, tea remains widely used in traditional practice. The concentration of menthol in tea is lower than in oil, but sufficient to stimulate receptors and produce mild relaxation. Peppermint tea does not contain caffeine. It does not stimulate the central nervous system in the way coffee or black tea does. Its sensory effect is more localized. Cooling in the mouth, softening in the stomach, aroma in the nose. The ritual itself plays a role. Water is heated, leaves are measured, steam rises, the cup is held. Breath slows naturally when sipping something warm. The act of preparing tea introduces a pause in activity. The nervous system responds to routine and repetition. Aroma, warmth, and slow intake combine. Over time, receptors adapt. The first sip feels most intense. Later sips feel softer. The brain recalibrates to the ongoing presence of menthol. What was sharp becomes gentle. This adaptation prevents overstimulation. It allows the experience to settle into the background. Peppermint tea also contains small amounts of flavonoids and other plant compounds. These contribute subtle antioxidant properties, though their concentrations in tea are modest compared to concentrated extracts. The leaves themselves remain intact after steeping. Their oil content reduced. Their structure softened. They release what they can, then rest at the bottom of the cup. Steam dissipates. Aroma fades slowly. Peppermint tea demonstrates how chemistry, temperature, and perception interact in simple ways. Heat extracts, vapor disperses, receptors activate, signals travel inward. The body remains unchanged in temperature overall, yet the sensation feels cooling. Warm liquid enters, cool receptors respond, muscle tension softens. In that small sequence, heat, diffusion, activation, peppermint expresses itself fully without force. From the ritual of tea, peppermint moves into a more measured environment. Clinical settings, laboratory studies, controlled dosages, where traditional use meets modern validation, and researchers attempt to quantify what experience has long suggested. For centuries, peppermint was used because people observed its effects. A cup of tea after a heavy meal, crushed leaves inhaled for clarity, oil rubbed lightly on the temples. The language used to describe those effects was simple: cooling, settling, fresh. In modern medicine, those observations have been tested more formally. Clinical trials attempt to measure what earlier generations described. Dosage is standardized. Placebos are used. Outcomes are recorded and compared. One of the most studied uses of peppermint in modern medicine involves irritable bowel syndrome, or IBS. Multiple randomized controlled trials have evaluated enteric-coated peppermint oil capsules. Meta-analyses, which combine results from many studies, have found that peppermint oil can significantly reduce abdominal pain and global IBS symptoms compared to placebo in some patients. The enteric coating plays an important role. Without it, peppermint oil might dissolve in the stomach and cause heartburn or irritation. With it, the capsule passes intact into the small intestine before releasing its contents. This targeted delivery allows menthol and related compounds to interact directly with intestinal smooth muscle. The mechanism appears to involve calcium channel modulation and smooth muscle relaxation. When excessive contraction contributes to discomfort, reducing that tension can ease symptoms. Peppermint has also been studied for tension-type headaches. In some controlled trials, topical menthol applied to the forehead and temples reduced headache intensity compared to placebo. The cooling sensation may interrupt pain signaling pathways or shift attention away from discomfort. Again, the effect is not absolute. It does not eliminate headaches entirely, but it may soften intensity in certain cases. Respiratory uses have also been examined. Menthol does not act as a decongestant in the traditional sense. It does not shrink swollen nasal tissue in a clinically measurable way. However, studies show that menthol can improve the subjective feeling of nasal airflow. Participants often report that breathing feels easier after inhaling menthol vapor, even when objective airflow measurements remain unchanged. This distinction between subjective and objective effect is important. Peppermint's strength lies in perception. It activates cold-sensitive receptors. It alters signaling. It changes how the brain interprets incoming sensory data. In cough drops, menthol provides a cooling sensation that may reduce throat irritation. In vapor rubs, it stimulates receptors on the skin and in the nose. The sensation of airflow increases, even though lung function remains stable. Safety considerations are part of modern evaluation as well. Peppermint oil is generally considered safe in small, controlled doses. However, high concentrations can irritate skin or mucous membranes. Undiluted essential oil should not be applied directly to sensitive areas. Internal use of concentrated oil should follow clinical guidance. Children and infants require particular caution. Menthol near the nose of very young children can sometimes trigger reflexive breathing responses. For that reason, many medical guidelines recommend avoiding strong menthol products in infants. Peppermint tea, by contrast, is much milder. The concentration of menthol is significantly lower than in oil capsules or extracts. For most adults, peppermint tea is well tolerated. Modern medicine often separates traditional use from evidence-based application. A practice that feels helpful is tested under controlled conditions. Sometimes the results confirm tradition. Sometimes they show only modest effects. Sometimes they reveal benefits in specific contexts rather than broad ones. Peppermint occupies an interesting space between tradition and validation. It is not a cure-all. It is not a replacement for prescription treatment in serious conditions, but its mechanisms are measurable. Its interactions with receptors are well documented. Cooling is not just a poetic description. It is a receptor-mediated event. Relaxation of smooth muscle is not simply anecdotal. It can be observed in controlled environments. And yet, the experience remains sensory at its core. Perception shifts. Breath feels clearer. Muscles feel cooler. The stomach feels calmer. From clinical studies, we move back toward one of peppermint's most familiar sensations: the feeling of open air moving through the nose, shaped not by structural change, but by receptor activation and neural interpretation. There is a moment when menthol first reaches the nose. A small shift. A subtle wave of coolness that seems to move inward with the breath. The air feels sharper, clearer, almost wider. Yet the structure of the airway has not changed. The nasal passages are lined with delicate tissue. Blood vessels beneath the surface can swell during illness or irritation, narrowing airflow. True decongestants work by constricting those blood vessels, physically reducing swelling. Menthol does not do this in a measurable way. Instead, it activates cold-sensitive receptors embedded in the lining of the nose. These receptors are the same TRPM8 channels described earlier. They respond to lower temperatures under normal conditions. When cool air passes through the nasal cavity, they open and send signals toward the brain. The brain interprets those signals as freshness and openness. Menthol binds to those receptors and activates them directly. Even if the air is warm, even if swelling remains unchanged, the signal is sent. The brain interprets it as increased airflow. Studies that measure nasal airflow objectively often show no significant change after menthol inhalation. Air volume remains consistent. Passage diameter remains stable. But subjective reports tell a different story. Participants frequently describe breathing as easier and clearer after exposure to menthol vapor. This difference highlights something important about sensory systems. Perception does not always require structural change. It requires receptor activation. Air moving across activated cold receptors feels different from air moving across inactive ones. The contrast between warmth in the lungs and coolness in the nasal passages enhances the sensation. Warm air enters the chest. Cool signals travel from the nose. The brain integrates both. The result is an illusion of expanded space. Menthol's volatility contributes to this experience. When a peppermint lozenge dissolves in the mouth, menthol vapor rises with each exhale. It reaches the nasal cavity from below. Inhaled vapor rubs and aromatic balms release menthol directly into the surrounding air. With each breath, molecules contact the lining of the nose. The effect is strongest at first exposure. Receptors activate quickly. Signals surge. Cooling feels immediate. Over time, receptors adapt. The intensity fades. What once felt sharp becomes subtle. This adaptation is protective. Continuous activation would overwhelm the system. Instead, sensitivity decreases with prolonged exposure. Peppermint appears in many products designed for respiratory comfort. Cough drops contain menthol to soothe throat irritation and enhance airflow perception. Inhalers and chest rubs rely on menthol's ability to stimulate cold receptors without altering airway structure. Even toothpaste uses menthol to create a sense of freshness that extends into the breath. Mint does not chemically remove odors from the airways. It does not eliminate bacteria directly in most formulations. It overlays its own signal onto the sensory field. The brain prioritizes the stronger input. Freshness becomes a neurological interpretation. The same mechanism explains why menthol can feel cooling even in hot environments. When cold receptors activate, the brain interprets the input based on signal patterns, not on measured temperature. Cooling sensation arises from electrical impulses along sensory nerves. Peppermint's relationship with breath is therefore perceptual rather than structural. Air moves as it always does. The diaphragm lowers, the lungs expand. Oxygen diffuses across membranes into blood. Carbon dioxide leaves in return. Gas exchange remains governed by physics and chemistry. Menthol does not alter that exchange. It alters the experience of it. Cool receptors fire. Signals travel along trigeminal nerve pathways. The brain assembles meaning, clear, open, fresh. And for a brief time, the breath feels easier. From this internal sensation of airflow, peppermint's influence extends outward again, toward the skin and muscles, where cooling can distract from deeper sensations and shift awareness in different ways. When menthol touches the skin, the sensation arrives quickly. A thin layer of cream spreads across the surface. Within seconds, coolness begins to bloom outward from the point of contact. It feels as though temperature has dropped, but the skin itself remains nearly the same. The cooling begins in receptors. Menthol activates TRPM8 cold-sensitive channels embedded in sensory nerve endings just beneath the surface of the skin. These receptors send signals along peripheral nerves toward the spinal cord. The brain interprets those signals as cold. But menthol's influence does not stop there. In sports, creams, and topical balms, menthol is often used as a counter-irritant. A counter-irritant works by creating a mild, non-harmful sensation that distracts from deeper discomfort. When cooling receptors activate strongly, they compete with pain signals traveling from sore muscles beneath the skin. The nervous system prioritizes the more immediate input. Cooling becomes the dominant sensation. Ache softens in awareness. This does not mean the muscle tissue has changed structurally. Menthol does not repair torn fibers or reduce inflammation directly in the way anti-inflammatory medications might. Instead, it alters the way signals are processed. Sensory information travels along specific nerve fibers. Pain signals often move through slower C fibers and faster A delta fibers. Cooling signals from TRPM8 receptors move through similar pathways. When multiple signals arrive simultaneously, the spinal cord and brain integrate them together. The concept is sometimes described through the gate control theory of pain. According to this framework, non-painful stimuli can partially close the gate to painful input at the spinal level. When cooling input is strong enough, it reduces the intensity of perceived soreness. Menthol may also interact with TRPA1 and TRPV1 receptors, which are involved in sensing heat and irritation. At certain concentrations, menthol can create a shifting sensory pattern. First cool, then mild warmth. This contrast arises because the nervous system recalibrates as receptor activation changes. In many sports balms, menthol is paired with other compounds that stimulate warmth receptors. The combination produces alternating sensations. Cool spreads first, then warmth follows. The sequence increases blood flow to the surface of the skin. The person perceives circulation improving. The increased blood flow perception does not necessarily reflect deep muscle repair. It reflects changes in skin blood vessels responding to sensory input. Athletes often use menthol creams after exercise. The cooling can feel refreshing on fatigued muscles. It can provide temporary relief from delayed onset muscle soreness. Studies suggest that menthol may increase pain threshold slightly when applied topically, though the effect is modest. Peppermint oil diluted properly can serve a similar function. Undiluted oil should not be applied directly to skin because of its concentration. When diluted in carrier oil, peppermint oil can provide a cooling sensation comparable to commercial preparations. The skin responds to menthol as it does to cold air. Blood vessels near the surface may first constrict slightly, then dilate as the body compensates. This vascular response contributes to the alternating cool and warm feeling that sometimes follows application. The effect remains temporary. As receptors adapt, cooling fades. The brain recalibrates. Soreness returns to baseline awareness. Peppermint does not override the body's repair process. It overlays sensation during that process. Cooling shifts attention. Attention shifts perception. The surface of the skin becomes the focus, rather than the deeper muscle fibers beneath. From receptor activation to nerve transmission, the pathway remains consistent with earlier sections. A molecule binds, a channel opens, ions move, a signal travels, perception forms. On the skin, as in the nose and tongue, menthol demonstrates how experience can be shaped without altering structure. Cooling rests on the surface. Signals travel inward. And from this external application, attention moves outward again, beyond the individual body, into fields of green leaves growing in temperate soil, where peppermint begins long before it reaches a tube of cream or a bottle of oil. Before peppermint becomes oil, tea, or cream, it grows low to the ground in wide green fields. From a distance, peppermint farms appear as smooth carpets of leaves, moving gently in the wind. The plants rarely grow tall. Most remain under 2 feet in height, branching outward more than upward. Peppermint prefers moist soil and temperate climates. It thrives in regions where summers are warm but not extreme, and where soil retains enough water to support steady growth. Too much heat can reduce menthol concentration. Too little sunlight can limit oil production. In the United States, large-scale peppermint cultivation is centered in Oregon and Washington. The Pacific Northwest offers long summer days and consistent soil moisture. These conditions encourage high menthol content in the leaves. The balance of temperature and light affects how the plant produces its aromatic compounds. Peppermint grows from rhizomes rather than seeds. Farmers plant root cuttings in early spring. These underground stems spread horizontally, sending shoots upward as soil warms. Because the plants are clones, fields grow uniformly. Each plant carries the same genetic traits as the one before it. During the growing season, farmers monitor height and leaf density. Oil concentration increases as the plant approaches flowering. Just before full bloom, menthol levels typically peak. This timing is critical. Harvesting too early may reduce yield. Harvesting too late can alter the oil's chemical profile. When the crop is ready, mechanical harvesters cut the plants close to the ground. The leaves and stems are left briefly in rows to wilt. This wilting reduces water content slightly, making distillation more efficient. After wilting, the plant material is gathered and transported quickly to distillation facilities. Timing remains important even here. The longer harvested peppermint sits before distillation, the more volatile compounds may evaporate into the air. Inside the distillation unit, steam rises through chopped leaves. Oil vapor separates and condenses. The extracted oil is stored in large containers, often stainless steel, to preserve purity. Peppermint agriculture requires careful soil management. Because the plant spreads through rhizomes and remains in the ground year after year, farmers practice crop rotation. After several seasons, peppermint fields may be rotated with grains or other crops to prevent soil nutrient depletion and reduce pest buildup. Soil health directly affects oil quality. Nutrient balance influences leaf growth and menthol synthesis. Excess nitrogen can encourage rapid leaf growth, but may dilute oil concentration. Farmers adjust fertilization to maintain the right balance between biomass and oil potency. Irrigation systems provide consistent moisture during dry periods. Too little water stresses the plant. Too much water can promote root disease. The goal is steady growth, not extremes. Peppermint fields also attract pollinators during flowering, though seed production is minimal due to sterility. The small purple flowers still offer nectar. Bees move among them. Even sterile hybrids participate in the broader ecological system. Weather plays a defining role in oil yield each year. A cooler growing season often produces oil with higher menthol content. A hotter summer may reduce menthol slightly and increase menthone. Subtle chemical shifts occur depending on environmental conditions. Peppermint agriculture is both biological and mechanical. Rhizomes extend beneath soil. Machines cut and gather leaves above it. Steam transforms plant matter into concentrated oil. Fields are replanted every few years as productivity declines. Old rhizomes are removed. Fresh cuttings are inserted. The cycle begins again. From sunlight to chlorophyll, from carbon dioxide to menthol, the plant converts energy into aromatic chemistry. That chemistry accumulates inside tiny oil glands until harvest releases it. Cooling begins not in the bottle, but in the field. Low leaves absorb light. Roots spread quietly underground. Farmers track timing carefully. And from those temperate fields of green, peppermint moves outward beyond regional farms, entering global trade networks where demand for menthol shapes cultivation across continents. Peppermint may grow low in a single field, but its influence stretches far beyond any one region. What begins as a patch of green leaves in temperate soil becomes part of a global network of cultivation, distillation, trade, and manufacturing. Cooling has become an international commodity. The United States, particularly Oregon and Washington, produces a significant share of high-quality peppermint oil. The climate in these regions supports strong menthol content. Long daylight hours during summer allow plants to build oil reserves steadily before harvest. But the global center of menthol production today is India. India grows large quantities of mint varieties specifically cultivated for high menthol yield. Through selective breeding and optimized distillation techniques, Indian producers supply much of the world's menthol crystals and mint oil used in flavoring, cosmetics, and pharmaceuticals. The climate in northern India differs from the Pacific Northwest. Summers can be hotter. Soil composition varies. These differences influence the chemical profile of the oil. Menthol concentration, menthone levels, and minor compounds can shift depending on region. Oil from one country may smell slightly sharper or sweeter than oil from another. Chemists analyze peppermint oil through gas chromatography to measure its exact composition. Small variations in menthol percentage affect how strongly the oil activates cold receptors. Even a few percentage points can alter the sensory intensity. Climate plays a major role in these variations. Cooler nights often promote higher menthol content. Excessive heat can shift the balance toward menthone or other related compounds. Soil nutrients and irrigation practices also influence oil chemistry. Because peppermint spreads through rhizomes rather than seeds, farmers worldwide often cultivate cloned varieties selected for consistency. Fields in one country may share genetic lineage with fields thousands of miles away. Once distilled, peppermint oil enters international markets. It is transported in large metal containers to manufacturers. Some of it is fractionated further to isolate pure menthol crystals. These crystals are used in cough drops, toothpaste, topical creams, chewing gum, and countless other products. The flavoring industry relies heavily on peppermint and menthol. Cooling compounds are used not only for mint flavor, but to enhance the perception of freshness in oral care products. The economic scale is substantial. Thousands of tons of peppermint oil and menthol are produced annually across the globe. Global demand shapes cultivation patterns. When demand rises, more land may be dedicated to mint crops. When synthetic menthol production increases, agricultural markets adjust. Synthetic menthol, produced chemically rather than extracted from plants, now supplies part of the global demand as well. Even so, natural peppermint oil remains highly valued for its complex profile. The subtle balance of menthol, menthone, and menthol acetate in plant-derived oil often differs from synthetic isolates. Trade routes that once carried dried herbs now carry refined oils. Containers cross oceans. Quality testing ensures purity, and safety standards are met. Cooling sensation, once confined to a leaf crushed in a garden, now moves through global supply chains. Peppermint's journey from field to consumer spans continents. Farmers monitor harvest timing. Distillers capture volatile oils. Chemists verify composition. Manufacturers incorporate menthol into products designed for flavor, scent, and sensation. Despite the scale, the underlying mechanism remains unchanged. Menthol molecules interact with TRPM8 receptors in the same way, whether grown in Oregon soil or Indian fields. Climate shapes chemistry. Chemistry shapes perception. From regional fields to international trade, peppermint reflects how a plant's natural properties can align with human sensory systems in ways that support widespread demand. Cooling travels across borders. Aroma disperses through air. Oil moves through markets. And from global trade networks, peppermint returns to everyday life again, not only as oil and medicine, but as a flavor paired with sweetness, bitterness, and fat in foods across cultures around the world. Cooling changes when it meets sweetness. Peppermint on its own is sharp, clear, almost piercing at high concentration. But when paired with sugar, chocolate, or cream, the experience shifts. The cooling remains, yet it feels smoother, balanced, integrated into something richer. There is chemistry behind that pairing. Peppermint's primary compound, menthol, is volatile and aromatic. When placed in food, especially foods that contain fat, those volatile molecules bind differently than they do in water alone. Fat carries aroma well. It slows the evaporation of certain compounds, allowing flavor to linger longer on the tongue. Chocolate contains cocoa butter, a type of fat that melts at body temperature. When chocolate melts in the mouth, it releases aromatic compounds gradually. If peppermint oil is added to chocolate, menthol dissolves into that fat. As the chocolate melts, menthol disperses smoothly across taste receptors. Cooling and sweetness meet at the same time. There is also a sensory contrast effect. Sweetness activates different taste receptors than cooling does. When two distinct sensory pathways are stimulated together, the brain interprets the contrast as heightened intensity. The coolness of mint can make sweetness feel brighter. The sweetness can soften the sharp edge of menthol. Contrast enhances perception. This may explain why peppermint is commonly paired with desserts. Peppermint bark, mint chocolate chip ice cream, candies. The cooling sensation creates a refreshing counterpoint to sugar and fat. In ice cream, the effect is layered. Cold temperature activates cold receptors naturally. Menthol activates them chemically. Together, they amplify the perception of coolness. Even a slightly warmer mint ice cream may feel colder than a non-mint flavor at the same temperature. Peppermint does not lower the freezing point of the dessert significantly. It changes how the cold is perceived. Peppermint also appears in savory cuisines. In Middle Eastern dishes, mint is added to yogurt sauces. The cool sensation balances spices. In Vietnamese and Thai cuisine, fresh mint leaves are paired with herbs, meats, and noodles. The volatile oils disperse quickly when leaves are torn, releasing fragrance into warm air. In savory contexts, peppermint or other mint varieties interact with salt and acidity rather than sugar. The cooling sensation contrasts with heat from chili peppers. The brain integrates both signals. Heat receptors respond to capsaicin. Cold receptors respond to menthol. The simultaneous activation produces a layered experience. Mint does not neutralize spice. It overlays cooling onto warmth. Peppermint's presence in food is often modest. Only small amounts are needed to trigger perception. Because menthol is potent, excess can overwhelm. Food scientists measure concentration carefully. Even slight changes alter the sensory balance. In baking, peppermint extract is added in drops. In confections, oil is diluted before mixing. The volatility of menthol means that aroma rises before taste arrives. The first impression is often scent. Peppermint can also influence how sweetness is interpreted. Some research suggests that cooling sensations may increase perceived freshness, which can enhance the enjoyment of sweet flavors without requiring additional sugar. Again, perception plays a central role. Mint does not increase sugar content. It adjusts how flavor feels. Across cultures, peppermint has become associated with seasonal desserts in some regions, particularly winter holidays. The contrast between cold outdoor air and cooling flavor reinforces the sensory theme. Candy canes, striped in red and white, carry the scent of menthol even before they are tasted. The pairing of peppermint and chocolate in particular has become iconic in many countries. The bitterness of cocoa and the sweetness of sugar are sharpened by the clean edge of mint. Cooling cuts through heaviness. Fat carries aroma. Sweetness balances sharpness. Cold receptors activate alongside taste receptors. From leaf to oil, from oil to confection, peppermint adapts to different culinary frameworks. It participates in dessert, in savory dishes, in drinks and candies. Its chemical properties remain the same. Menthol binds to receptors. Volatile molecules rise. But when cooling meets sweetness or spice, the experience expands. And from food, peppermint's role shifts once more, not as flavor alone, but as part of the plant's original biological strategy, where sharp aroma and cooling compounds functioned long before they met chocolate or sugar. Before peppermint was steeped in tea or distilled into oil, its strong scent served another purpose. In the natural world, aroma is not simply fragrance. It is communication. It is warning. It is defense. Plants cannot run from predators. They cannot hide easily. Instead, many produce chemical compounds that discourage grazing or insect feeding. Peppermint's sharp, cooling aroma likely evolved as part of that survival strategy. Menthol and related compounds can deter certain insects. When volatile oils are released from peppermint leaves, they create a chemical barrier in the surrounding air. For small insects that rely heavily on scent to locate food and navigate their environment, strong aromatic compounds can interfere with those signals. Some studies have shown that peppermint oil can repel ants, mosquitoes, and certain beetles under controlled conditions. The mechanism varies depending on species, but the general principle remains consistent. The intense scent disrupts normal behavioral patterns. It can mask pheromone trails. It can irritate sensory organs. It can create an environment that insects prefer to avoid. Essential oils in many plants function this way. Peppermint's oil glands store concentrated aromatic compounds precisely for release when leaves are crushed or damaged. When an insect begins to feed, the rupture of oil glands increases local concentration of menthol and other terpenes. The sudden release of scent may discourage further feeding. This chemical defense is not unique to peppermint. Many members of the mint family produce aromatic oils. Basil, oregano, lavender, and rosemary all contain volatile compounds that deter certain pests. Evolution favors plants that reduce herbivory. Over time, individuals producing stronger deterrent compounds may survive and reproduce more successfully. In peppermint's case, hybridization intensified menthol production. While this may not have been selected solely for insect deterrence, the high menthol concentration likely contributed to its resilience. Gardeners sometimes use peppermint oil as a natural repellent. Diluted sprays are applied around entry points to discourage ants or spiders. The effect is temporary and depends on concentration. But the strong scent can alter insect movement patterns. Companion planting also reflects this ecological role. Growing mint near certain crops may help reduce pest pressure. The volatile oils released into the air create a subtle protective zone. Again, this is not absolute protection. It is modulation of environment. Peppermint scent moves quickly because its compounds evaporate easily. This volatility makes it effective as a short-range deterrent. The plant invests energy in producing molecules that disperse into air and signal presence. Chemical communication is central to plant ecology. Some plants release compounds that attract beneficial insects, such as pollinators or predators of pests. Others release compounds when damaged that signal neighboring plants to increase their own defensive chemistry. Peppermint's aromatic profile participates in that larger system. To an insect, menthol may not feel refreshing. It may feel overwhelming or disorienting. Sensory systems in insects differ from those in humans. What activates human cold receptors may interact differently with insect chemoreceptors, but the intensity of volatile terpenes can still disrupt behavior. In this way, peppermint's cooling compound serves dual roles. To humans, it feels clarifying. To certain insects, it signals avoidance. The same molecule can carry different meanings depending on the nervous system interpreting it. Fields of peppermint may experience less insect damage than non-aromatic crops, though no plant is completely immune. Farmers still monitor for pests and manage fields carefully. Peppermint scent offers support, not invulnerability. Strong aroma rises from leaves in warm sun. Insects approach. Some turn away. From defense in the field, peppermint's chemistry moves inward again, not to muscles or digestion this time, but to the brain itself, where scent meets memory and perception in ways that extend beyond survival and into cognition. Scent reaches the brain differently than other senses. When light enters the eyes, it is routed through multiple processing centers before awareness forms. When sound enters the ears, it travels through relay stations deep within the brain. But scent follows a more direct path. When peppermint vapor enters the nose, volatile menthol molecules bind to olfactory receptors high in the nasal cavity. Those receptors send electrical signals to the olfactory bulb, a structure located just above the nasal passages at the base of the brain. From there, signals travel directly into parts of the limbic system, including the amygdala and hippocampus. These regions are involved in emotion and memory. This direct connection is one reason scent can feel immediate and vivid. It can trigger recollection without conscious effort. A hint of peppermint might recall a childhood candy, a winter holiday, a tube of toothpaste, or a cup of tea late at night. The brain links scent with context quickly. Peppermint has also been studied for its effects on alertness and focus. Some laboratory studies suggest that exposure to peppermint aroma can increase subjective feelings of alertness and reduce perceived fatigue. Participants performing simple tasks sometimes report improved concentration when exposed to mint scent. The mechanism is still being explored. Menthol activates not only cold receptors, but also trigeminal nerve pathways in the face. The trigeminal nerve carries sensory information related to touch, temperature, and irritation. Activation of this system can produce a mild stimulating effect. The sharpness of peppermint may increase arousal slightly compared to softer scents. This stimulation is gentle. It does not function like caffeine. It does not significantly increase heart rate or alter central nervous system chemistry in a strong way. Instead, it shifts sensory tone. The crispness of peppermint can feel clarifying. Context matters. In a quiet room at night, peppermint tea may feel calming. The warmth of the liquid, the ritual of preparation, and the gentle cooling sensation can support relaxation. In a bright workspace during the day, peppermint aroma may feel invigorating. The brain interprets scent within environmental context. Association learning plays a role here. If peppermint has been paired repeatedly with cleanliness, alertness, or study sessions, the brain may begin to anticipate those states when the scent appears. This is classical conditioning. The scent becomes a cue. Memory networks form connections between sensory input and emotional response. The hippocampus encodes experiences. The amygdala assigns emotional tone. Repeated pairing strengthens neural pathways. Over time, peppermint may come to represent clarity or freshness, not because of intrinsic stimulation alone, but because of learned association. Research also suggests that peppermint scent may modestly enhance mood in certain contexts. Some participants report feeling more energetic or less mentally fatigued when exposed to peppermint aroma. However, responses vary between individuals. Not everyone interprets peppermint the same way. Cultural background, personal history, and environment shape interpretation. For some, peppermint may feel medicinal. For others, festive. For others still, routine. The olfactory system adapts quickly. Continuous exposure reduces sensitivity. The first breath of peppermint feels strongest. Later breaths feel softer. The brain recalibrates to the ongoing presence of scent. This adaptation prevents overstimulation and allows new sensory input to stand out. Peppermint's interaction with the brain demonstrates how perception blends biology and experience. Menthol activates receptors. Signals travel along the olfactory nerve. The limbic system responds. Memory links engage. Cooling meets context. A single molecule can trigger both a sensory response and an emotional one. From scent reaching memory, peppermint's influence extends further into learned associations of freshness and cleanliness, shaped not only by biology, but by repeated exposure in daily life. Freshness is not a physical property. It is an interpretation. Peppermint has become deeply linked with the idea of cleanliness, especially in modern environments. Toothpaste, mouthwash, gum, soap, shampoo, surface cleaners, and even air fresheners often contain mint fragrance. Over time, repeated exposure has shaped a powerful association between peppermint scent and the feeling of being clean. The connection begins with sensory contrast. Menthol activates cold receptors in the mouth and on the skin. Cool sensations are often associated with water, air, and renewal. When brushing teeth with mint toothpaste, the cooling signal follows mechanical cleaning. The brain links the two experiences. Scrubbing removes debris. Menthol activates receptors. Cooling spreads. The sequence reinforces the idea that clean equals cool, even though the cooling does not chemically remove bacteria. In hygiene products, mint fragrance often masks other odors rather than neutralizing them completely. Yet the brain interprets the stronger mint signal as dominant. If unpleasant smells are replaced by peppermint aroma, the environment feels fresher. Repeated exposure strengthens the association. From early childhood, many people encounter mint during brushing routines. The scent appears every morning and night. The brain encodes the pairing of peppermint with hygiene habits. Over years, the link becomes automatic. Marketing has amplified this effect. Manufacturers deliberately incorporate mint into products to signal freshness. When consumers smell peppermint in a room spray or cleaning solution, they often interpret the space as sanitized, even if actual microbial levels remain unchanged. Research in scent science suggests that certain aromas can influence perception of cleanliness. In experimental settings, participants exposed to minty scents rated environments as cleaner compared to unscented spaces. The scent altered subjective judgment. Conditioning shapes perception. If peppermint is consistently paired with tasks that remove dirt or odors, the brain begins to treat the scent as evidence that cleaning has occurred. This phenomenon is not unique to mint, but mint's cooling sensation strengthens the effect. The physical activation of cold receptors adds a tangible component to the scent. It feels active rather than passive. Peppermint in mouthwash creates tingling in the mouth. That tingling reinforces the idea that something is happening. The sensation becomes proof of action. Even though antimicrobial ingredients may work independently of menthol. Over time, the scent of mint can influence how people evaluate spaces. A mint-scented room may feel more orderly or hygienic than one without scent. This perception arises from learned association rather than direct chemical evidence. Context remains important. In a kitchen, peppermint scent may signal cleanliness. In a dessert shop, it may signal flavor. In a clinic, it may signal sterility. The same molecule produces different interpretations depending on environment. The olfactory system's direct link to memory strengthens these associations. When peppermint activates receptors, the olfactory bulb sends signals into limbic regions tied to emotion and learned patterns. The brain retrieves stored meaning quickly. Cooling becomes symbolic. Peppermint's role in perceived cleanliness demonstrates how sensory experience shapes interpretation of the world. Menthol activates receptors. The brain registers coolness. Repeated pairing with hygiene routines builds a cognitive shortcut. Cool equals clean. The association becomes so strong that even synthetic mint fragrance can evoke it. Yet beneath the symbolism, peppermint remains a plant in the mint family. Its square stems and opposite leaves still define its botanical identity. Its oil glands still store volatile compounds for ecological purposes. From psychological association, attention turns back to the plant itself, to the structural traits that connect peppermint with its aromatic relatives in the broader mint family. Step back from oil bottles and toothpaste tubes, and peppermint becomes what it has always been: a plant. It belongs to a botanical family known as Lamiaceae, often called the mint family. This family includes many aromatic herbs used in cooking and fragrance: basil, lavender, oregano, rosemary, thyme, sage. Though their scents differ, their structures share clear similarities. One defining characteristic is the square stem. If you gently roll a mint stem between your fingers, you may feel edges rather than a smooth cylinder. The four-sided shape is common among members of the mint family. It is not accidental. It reflects patterns in how vascular tissue is arranged inside the stem. Leaves in the mint family typically grow in opposite pairs. Each set emerges directly across from the other along the stem. This arrangement maximizes light exposure while maintaining structural balance. Peppermint follows this pattern precisely. The leaves are bright green with serrated edges. Their surfaces contain glandular trichomes, the small oil-producing structures discussed earlier. Under magnification, these glands appear as tiny rounded protrusions. Inside them, aromatic compounds accumulate as the plant grows. When the leaf is crushed, the glands rupture. Volatile molecules disperse into air. This oil production is a shared trait across the family. Lavender produces linalool and linaloyl acetate. Basil contains eugenol and linalool. Oregano carries carvacrol and thymol. Each species synthesizes its own blend of terpenes and aromatic compounds. These compounds serve ecological purposes. They deter herbivores, attract pollinators, protect against microbial threats. The mint family is defined not just by structure, but by scent. Flowers in the mint family often share a similar shape. Many have bilabiate flowers, meaning the petals form two distinct lips. This structure guides pollinators toward nectar, while ensuring pollen transfer. Peppermint flowers are small and pale purple, arranged in clusters along the stem. Even though peppermint is largely sterile and does not produce viable seeds, it still forms flowers. These blooms offer nectar to insects and contribute to the plant's ecological interactions. The Lamiaceae family includes over 7,000 species worldwide. Many thrive in Mediterranean climates with warm summers and well-drained soil. Others adapt to temperate or even tropical regions. Aromatic oils are central to this family's identity. From a biochemical perspective, many mint family plants produce terpenes through similar metabolic pathways. These pathways convert simple carbon units derived from photosynthesis into complex aromatic molecules. Sunlight enters chloroplasts. Carbon dioxide is fixed into sugars. Enzymes reshape those sugars into terpenes. Those terpenes accumulate in oil glands. Cooling, fragrance, and flavor begin with photosynthesis. Peppermint's menthol content distinguishes it within the family, but the underlying process is shared. The mint family is a lineage of aromatic plants shaped by evolutionary pressures that favored scent as a survival tool. Gardeners often recognize mint family members by scent before structure. A single brush of the leaf releases aroma. Even closely related species can be identified by their distinct fragrance. Square stems, opposite leaves, oil glands along the surface. Peppermint fits perfectly within this botanical framework. Zooming back out reveals how a simple plant structure supports the chemical complexity explored throughout this video. The cooling sensation, the digestive relaxation, the aroma linked to memory, all trace back to glandular trichomes on opposite leaves attached to square stems. Botany supports chemistry. Chemistry shapes perception. And as we move toward the final descent, attention returns not to fields or families, but to the quiet sensation where this journey began. Cooling touching receptors, breath moving slowly, scent softening as awareness drifts toward rest. At the beginning, there was a simple sensation, a cool touch across the tongue, a thin ribbon of freshness moving through the nose, a leaf releasing scent when brushed by a hand. Menthol meets a receptor. A channel opens. A signal travels. The brain interprets it as cold. Not because the air changed, not because temperature dropped, but because chemistry fit into biology in just the right way. Cooling was always perception. Breath continues now, the same way it always has. In, out, slow and steady. Air moves across the nasal passages. Receptors respond less intensely than they did at first. Adaptation begins. What felt sharp softens. What felt vivid becomes gentle. This is how sensory systems protect quiet. When stimulation remains constant, awareness fades slightly. The nervous system recalibrates. Intensity gives way to neutrality. Peppermint demonstrates this clearly. The first breath of mint feels bright. The second feels lighter. By the third or fourth, cooling settles into the background. The sensation dissolves not because it disappears, but because perception adjusts. Breath keeps moving. The diaphragm lowers. The lungs expand. Air enters and leaves without effort. Menthol does not change that rhythm. It briefly colors it, then it fades. In the same way that scent rises from a cup of tea and slowly disperses into the room, cooling sensation disperses across awareness. It does not need to be held. It does not need to be maintained. It arrives. It activates. It softens. Throughout this journey, peppermint has moved through fields, through distillation chambers, through digestive muscle and skin receptors, through ancient gardens and modern laboratories. Yet each step returned to something small and precise. A molecule shaped in a leaf, a receptor waiting in a nerve ending, a signal interpreted in the brain, cooling without temperature change, freshness without structural shift, relief through modulation rather than force. Now, there is no need to analyze it further. Simply notice the breath. Notice how air feels against the inside of the nose. Notice how it no longer feels sharply cool, only neutral. Perception has settled. The scent, if it lingers in your mind, becomes faint, then softer still. Signals quiet. Awareness drifts. Cooling becomes stillness. Stillness becomes rest. And as the sensation dissolves completely into the background, nothing needs to be added. The breath continues. The receptors remain quiet. The mind follows. Good night.